Hi, everyone. Welcome to the Tuesday seminars on the cerebellum. Today we have as our guest Elina Nagaeva. She's a postdoc at the Translational Neuroscience Group at the University of Helsinki, Finland. She's an electrophysiologist and a neuroscientist by training. Her main research interests are in cell type specific physiology and pharmacology, with a focus on the monoamine system of the mammalian brain. She's going to be talking to us about the cerebellum today and the role of GDNF in regulating circuit function. She is a postdoc in the laboratory of Jean-Ole Andresu, who is also with us, and he can help with answering questions if time allows. Elina, thank you so much for being with us and presenting your work.
Hello, everyone. Yeah, thank you for the invitation. I'm very glad to be able to present our data to this community. And I want to I appreciate that you are all deeply interested in the cerebellum, and our lab is more like investigating the neurotrophic factors and the GDNF in particularly. And as I like to joke, well, we didn't go to the cerebellum, but cerebellum came to us. And today I will tell you on how the motor learning can be regulated by the GDNF levels in Purkinje cells. So what is the GDNF? GDNF is a small protein molecule which needs to like GFR1 receptor binding and then binding to the red receptor and their dimerization, which are themselves the tyrosine kinases, in order to produce its action on the cells in the body and the brain. And it regulates the proliferation and survival of many neurons, and not only. And it's historically was it's called the glial cell line derived neurotrophic factor, or GDNF. It's a historical name, but actually it's produced not like in many cells in the body and the brain. And it's majorly known as the survival factor for the dopamine neurons, but it's actually produced and acts in many other tissues, so including the muscles, and it's regulate the development of kidney, for example, and the gut neurons, and also the monoamine systems such as dopamine, as I mentioned, and the serotonin systems. So, but because like it's known, its effect on the dopamine system is pretty pronounced. It was tested as a treatment for the Parkinson's disease, for example. In particular, its ectopic delivery. But so far it wasn't very successful, unfortunately, because through the ectopic delivery, it's hard to regulate the concentration of GDNF, and when you apply too much of this molecule, then you have this like massive sprouting of the dopamine axons, and that's not always a good thing to have. Therefore, researchers are searching for like how to upregulate this molecule in controllable manner, and our lab was also developing these genetically modified animals, or called GDNF Hypermorph mouse model, which has like edited GDNF gene, and particularly it's after transcription, it has several modifications in the 3' UTR end of the gene. And after transcription, it lacks the binding site for the microRNAs. So basically, the stop signal is downregulated for this molecule. And as the result, we have the upregulation of GDNF protein in these animals. And we tested expression of GDNF mRNA in the different sites. This is for brain. And we found the like somehow threefold, threefold elevation in the dorsal striatum, where it's known to be produced, and in the cerebellum, which is interesting for us, and the spinal cord, and also other regions. But the most massive upregulation we observed in the striatum, and we confirmed that also at the protein level. So the protein levels was also upregulated twofold in the striatum in these animals. And because we know about it, like profound dopamine upregulation by the GDNF, we tested the concentration of the dopamine within the striatum. So basically it's coming to the striatum shop substantia nigra, but this is for dorsal striatum, yes. And we saw the upregulation of dopamine and as well as the higher dopamine release in the striatum. This was done by cyclic voltometry experiments. And therefore, like we were expecting to have some motor responses or some changes in the motor function of these animals, like particularly like whether they hyperactive, first of all. And we tested these in the open field experiment, and surprisingly, we didn't see the difference. So these animals were completely normal in sense of the open field exploration. But we found differences in the motor performance in the rotor-rod test at the first day. So basically they were performing better. And also this upregulation was staying on the second day. So basically we see some some improvement in the motor performance. And what is important for like maybe Parkinson's disease as well, and so that this improved motor performance, and learning actually was staying in adult animals as well. So not adult, old animals. They were 15, 17 months old, and like in the rotor-rod test, we didn't see any more the upregulation of the motor performance, but we saw the like better learning in these animals. So they were able to learn better. And then in the vertical grid test, they were better also on the day one. So they probably had better proprioception, or like they performed better, and they learned also better. But the thing is that motor function can be regulated by GDNF at several sites, including the central part of the motor system and the peripheral part. And this includes the, as I mentioned, nigrostriatal dopamine system, so substantia nigra, to the striatum and the motor cortex, the cerebellum, which like some cells express GDNF there, and there are receptors for GDNF. The Clark's column of the spinal cord, dorsal root ganglia, and peripheral skeletal muscle tissue, because GDNF is produced by muscles and it supports the survival of motor neurons. So where exactly? Basically, this paper, which I'm presenting today, was trying to narrow down the place where this GDNF upregulation is important for motor learning. And for that, we used different, basically genetic approaches. And we developed this conditional GDNF hypermorph allele called C-hyper, which helping to control the GDNF upregulation by like in time and in the cell type. So basically this mouse can be crossed with any Cree mouse line. And yes, and first of all, we wanted to see whether the central part of the motor system is important the most or not, or is the peripheral. So that's why we crossed these animals with a nesting Cree mouse line, which would, while crossing with conditional hypermorph GDNF mice, would restrict the expression, the upregulation of GDNF only to the cells which are naturally produce GDNF, but only in the central nervous system. And this upregulation happening at embryonic days 11 and 12. And we tested these animals in the rotor-rod, and we saw again no improvement in the rotor-rod performance, but improvement in the learning on the day 2, and improvement in performance in the vertical grid, and improvement in learning at this test as well. So basically, this was done in animals of 2-3 months old, so young adults. And we concluded that is studying of the overexpressing GDNF in the central nervous system at embryonic 11 day 11 and 12 is enough to produce this improvement in motor learning. Then another thing that still, even in the central motor system, there are different places where the receptors for GDNF are expressed, and this is called red receptors, I showed in the first slide. And like it's expressed in many places, like in the well, yes, in the substantia nigra, in the striatum, in the substantia nigra, in the red nucleus, in the cerebellum, in Purkinje cells, and in granular cells. Now we know it's a bit better, and also in pons, in pontine nucleus, and in different motor nuclei. So then we need to narrow down it even further. And because like striatum has the highest expression of the GDNF, and because we also wanted to kind of test it in more maybe applicable way. So basically we expected that the striatum would be the important place for the motor learning and performance. And yes, we right away tried to upregulate it in the adult animals. So it would probably have like more like clinical meaning, possibly. And we expressed then in these conditional hypermorph animals, when they were 2 months old, we expressed the virus, injected AAV virus holding CRE, and produced the overexpression of GDNF specifically in the striatum. And although we saw the increased concentration of the GDNF protein in the striatum, we didn't see any improvement in the motor learning or performance in these animals. So we concluded that probably this is not the most important site for this improvement. So we concluded that adult striatum GDNF upregulation is not involved in that. Then but because we had this proprioception component and also motor learning, so of course, it indicates that maybe cerebellum is important for that. And we tested in our constitutive and those which have upregulation in GDNF in all cells in the body and the brain. We tested basically we patched the Purkinje cells in these animals and recorded for the excitatory postsynaptic currents and inhibitory postsynaptic currents coming to Purkinje cells. And we saw that there was upregulation in the excitatory postsynaptic currents, and it was like the more pronounced in the homes. And similarly, we saw the decrease in the inhibitory postsynaptic currents, and it was pretty drastic. But we couldn't, these animals were P10 or P18, so because they actually, homozygous animals, they die at P25, so it's like 100% of animals die because of the kidneys problems, kidney malformation. So you can appreciate that this image, this is like how it develops. This is a normal kidney and urogenital basically development normal, but this is in the GDNF homozygous hypermorphs. This is the size of kidney that they get at P7, so it's probably very hard to live with such kidneys. Therefore, we wanted to do the upregulation specifically in PCP in Purkinje cells, basically because this is the major expression site of GDNF in the cerebellum. Also granule cells express, but in the lesser amount. And because Purkinje cells are important for motor learning and for granule cells too, but yes. So we did these PCP3 conditional hypermorph animals, GDNF hypermorph animals, which has upregulation. So the upregulation starts at the first postnatal week in Purkinje cells at around P4. And then these cells started to produce a bit more of GDNF than usually. But this you see here, it's not like drastic, but it's around twofold more GDNF. And we tested also in this striatum to confirm that it was specific to cerebellum, and we didn't see any upregulation of striatum. And we tested these animals similarly in the rotarod and the vertical grip test. And we saw that now these animals do not perform better in vertical grid and neither in the rotarod, but they actually learn better during the second day for each test. So they perform better second day rotarod and better in vertical grid. And these animals were around 2 to 3 months old. And basically, upregulation in the Purkinje cells is enough to produce this improved motor learning. Importantly, we of course tested whole other motor tests, and we didn't see any differences in these animals. So no difference in open field, how they moved, no difference in anxiety, and no difference in the multiple static rod test. And similarly, we tested beam walking and the grip strength, because for the vertical grip, probably it's kind of important thing, that how strong they are, and the same in the coat hanger test. So they were not different in other motor tests. So then we wanted to assess what happens at the circuitry level and the cerebral cortex, and we again recorded same EPSPs and EPSCs and iPSCs in the Purkinje cells. So yeah, just again to clarify, we upregulate GDNF in Purkinje cell, and we're recording Purkinje cell because we expect to have the circuit changes because there are no receptors for GDNF in the Purkinje cell, but in around neurons there are receptors. So we saw again the increase in the excitatory postsynaptic currents coming into the Purkinje cells, and we also saw with immunohistochemistry, we assessed whether there are more of VGLUT1 and VGLUT2 coming in innervation to the Purkinje cell layer, and we saw increase in both, in VGLUT1 and VGLUT2. And we assessed the inhibitory postsynaptic currents, and we didn't see any difference in that. It was a bit surprising because quite a lot of red receptors expressed in the basket cells, and we kind of were thinking that there should be some differences, but we didn't see any. Probably some compensation happened. Then, like, logically, we assessed how the spontaneous action potentials frequency changed in this, because we have more excitatory inputs coming, and we expect to see the upregulation of frequency. And that's indeed what we saw, basically, yes. And we saw the, like, around twofold. So they fire faster now, these Purkinje cells. But it was done without like blocking anything, so it's includes all the common innervation, of course. And it's good to point that this is basically situation opposite to some cerebellar ataxia, when we saw the decrease in the cell firing of the Purkinje cells. So yeah, and we see the improvement in motor learning. So we came up with this summary scheme. It's a bit complex, but probably for the cerebellar community, not so much. And yes, so there are receptors. This is a normal brain. We have mossy fibers and granule cells coming, and we have inferior leaves, VGLUT2 positive endings ending up in the Purkinje cell. And it was shown previously that the red receptor is expressed in the inferior leaves, and the red receptor is expressed in the pontine nucleus, for example, which form these mossy fibers. But in the end, we saw like basically upregulation in both. We saw upregulation in VGLUT2 synapses, probably through direct upregulation through the red signaling. And we saw the upregulation of VGLUT1 synapses, which kind of can't be direct. So it might be so that we have the upregulation like through the red signaling in these camin mossy fibers. And then we have more excitation from them. And then we produce more VGLUT1. But we, of course, didn't confirm that. But this is just a summary scheme. And this all like more excitatory postsynaptic currents causes the higher excitation of the Purkinje cell and the higher cell firing frequency of these cells, and associated with improved motor learning. We also like tested the human expression in the available data sets of this molecule, and this was the data set, so it's like Affymetrix assay, and this includes 120 samples which passed, like which had ring factors higher than 5, and we estimated like the basically log2 expression of these, yes, you can see here, and the fold change of GDNF, basically the range of the GDNF in healthy human cerebellum is around 2 folds. So like exactly what we saw in our mice. These kind of have like can provide a basis that it might be like our improved motor learning in mice can be aligned also with human abilities to learn some motor functions. Yes, and another important thing which our paper shows is that with no GDNF in Purkinje cells, motor learning is not affected. So we did these knockout animals, specifically in the Purkinje cell. So now we don't have any GDNF in Purkinje cell. Because, so this is the GDNF mRNA levels fold change in the whole cerebellum tissue. As we see here, we don't see the full deletion of GDNF because there are granule cells still which express it. And but in the nesting tree, this is the black, we don't have GDNF expression at all. And we assessed these animals also in the rotarod and in the vertical grid, and we didn't see any changes. So they were performing similarly as the wild type littermate control, like is the littermate controls. So basically, upregulation of GDNF in Purkinje cells is good for motor learning, but no GDNF in Purkinje cells probably compensated somehow by other cells, and it doesn't cause any drastic consequences for motor learning. So yeah, the conclusions that I've just said is that the postnatal twofold increase in GDNF levels in Purkinje cells improves motor learning. Deletion of GDNF have no consequences for motor performance or learning when we delete only from the Purkinje cells. GDNF upregulation in Purkinje cells enhances the excitatory innervation by boosting VGLUT1 and VGLUT2 containing synapses, resulting in an increased firing frequency of Purkinje cells. And as the major conclusion is endogenous GDNF level in postnatal Purkinje cells is an important but non-essential regulator of motor learning. And variation of GDNF levels in healthy human cerebellum may correspond to individual differences in motor learning abilities. Thank you for your attention.
Thank you, Elina. I'm happy to allow anybody who has questions to please just go ahead and raise your hand, and you can have an opportunity to have Elina explain it. So I will start, Elina. I was going to ask you about the relationship between the function of GDNF, dopamine, and whether there could be any relationship between how dopamine is acting in the cerebellum with regard to an upregulated GDNF?
Okay. Yes. So like basically if we're talking about the nesting tree animals, which have like upregulation everywhere, like and also not only in PCP, but also but sorry, are they coming to the Purkinje cells, the dopamine projections?
That's my understanding, that there are dopamine projections to the cerebellum.
Yeah, okay. And like, yeah, sorry, but I can't really answer this question because I don't know how they control the motor system. Jan-Ole, do you have anything in mind on that?
Yeah, hi there, and thanks for getting us to present. Yeah, but honest answer is I don't know. If you think of where dopamine can be reliably seen, then even in prefrontal cortex, where it's very important for cognition and various functions we use at a daily basis, dopamine cannot be really measured with cyclic voltammetry and analyzed properly. You can, and in cerebellum, if there I think is few fibers coming in, but they're not easy to functionally follow through. Probably not possible with current. So, but we cannot exclude it that if there is red positive dopamine fibers actually coming in, that they do react.
They should.
What is, I don't actually know what is a dopamine receptor situation on Purkinje cells or basket cells. We should be checked. I think this is an interesting point of view. Let's look at the single cell sequencing data, what dopamine receptors are out there.
Oh, wonderful.
Hi, thanks for such an interesting talk. I'd like to ask a question. You showed that the increased GDNF caused an increase in the synaptive connection with both the mossy fibers and the climbing fibers, if I'm understanding that correctly. I was wondering if you, when you showed that the Purkinje cell action potential rate increased, did you look at the difference between simple spikes and complex spikes? Just seeing the increases in motor learning, I'm wondering if there was any difference in how the Purkinje cell responds to a climbing fiber input.
Yes, thank you for your question. I understand what you mean. And yeah, so basically I tried to assess it in the experiment. I performed that, but I couldn't really see the complex spikes there. So maybe it's due to the like how I cut it, the cerebellum, like so I cut it out. Basically in many slides I had just the like cerebellum without like olives, and yes, so I tried to look for them, but I didn't see any. So for that I can't really say. So we address it as like changes in the simple spikes.
Thanks.
Elina, what are your plans for future studies?
Yes, thank you. So basically, it would be interesting to see what's going on in the development, because like cerebellum undergo quite like interesting development phases and patterns, and like it's for me it's quite unclear why we do not see any changes in the iPSCs, because yes, like it's quite a lot of red receptors there, and the iPSCs controls very strongly the output of the Purkinje cells, and because in these hypermorph mice, if you remember, we saw when they were young, so like P7, P14, we saw the decrease in iPSCs. And why do we not see it in the adults, in the conditional mice? It's kind of interesting question. And what's going on during like during the development? Why this more innovation coming? So yes, and the other like potential of this model, like it's kind of might be possible to cross it with ataxia model mouse line and to see whether we can improve their situation while they maturing through this upregulation in GDNF, either like with PCP2 specific upregulation or like in more general nesting tree mice.
Yeah, that would be pretty exciting if you can use this to rescue the ataxia mice.
Yes, that would be nice.
Wonderful. Are there any questions from the audience? Well, Elina, thank you so much for taking your time. Good to see you.
Yeah, thank you for inviting us, and good luck with your research.
Thank you. Bye. Bye,
Bye-bye.
everyone.